Okay, in this video we're going to talk about smooth muscle. Now we know that smooth muscle is found in the walls of hollow organs like your digestive tract or genitourinary tract or respiratory tract, except for the heart because the heart contains cardiac muscle. In terms of the microscopic structure of smooth muscle, we know that it's kind of spindle shaped. There's short, thin cells that only have one nucleus and no striations. Now, connective tissue is mostly lacking within smooth muscle. And so we find that it actually contains endomysium just to nourish those cells. Now, the smallest blood vessels of your body pretty much don't contain smooth muscle in their wall, but pretty much the rest of your blood vessels do. And it turns out, though, that the muscle that you find in hollow organs is usually arranged within two major layers. And the layers here have cells that basically oppose each other in perpendicular directions. We're going to have a circular layer and a longitudinal layer, and these actually allow for peristalsis, where when one layer contracts in one direction, another layer can contract in the other, and that allows for a wave of muscular contraction that propels, a substance in a particular direction. So if we look at the two layers of smooth muscle within the wall of our digestive tract, we find that there's a circular layer that actually surrounds the lumen, or the space within this digestive organ. Then we have a longitudinal layer that goes along its length, like lengthwise, longitudinally. And so if these two layers contract together, we actually get a peristaltic wave. So imagine a circular layer that would squeeze and a longitudinal layer that kind of propels and pushes along its length or shortens. Taken together, it actually is a wave-like motion. And so you get these undular motions, which we call peristalsis, that help to propel digestive contents along the length of your GI tract. So we actually find out here that smooth muscle doesn't have neuromuscular junctions. Instead, we actually have autonomic nerve fibers that innervate smooth muscle, and these actually contain what we call varicosities, which are these little blebs or pouches of vesicles that can actually release neurotransmitters directly onto smooth muscle. And so what these varicosities look like is with our nerve fiber here, we have these little blebs, and each bleb is a varicosity. Each varicosity is full of mitochondria, which supply the ATP to help for exocytosis of these vesicles containing neurotransmitters. Now, there are several different neurotransmitter types that can be released onto smooth muscle, whereas with skeletal muscle, it was always acetylcholine that's released at the neuromuscular junction. So smooth muscle doesn't contain sarcomeres. It doesn't contain myofibrils either, for that reason. And it doesn't have T-tubules. Instead, what we find here is that the sarcoplasmic reticulum of smooth muscle is far less developed. We see that it doesn't really store a whole lot of calcium. Rather, smooth muscle relies on the calcium from extracellular fluid. And this calcium comes from these little infoldings of the membrane called calveolae. Now, calveolae contain numerous voltage-gated calcium channels that allow for a rapid influx of extracellular calcium. So looking at a smooth muscle here, we see that there's kind of a spindle shape. We have one central nucleus, these infoldings called calveolae, which actually have voltage-gated calcium channels, and then gap junctions that allow for these cells to spread their action potentials from cell directly into the other cell. Now, you find that there are no myofibrils within a smooth muscle cell because they don't contain sarcomeres. And instead, what we find are these dense bodies that are linked up with the intermediate cytoskeleton inside of the cell, and these actually allow for contraction of the cell, and because they crisscross, it actually makes the whole cell kind of bunch up together. So the thick filaments are fewer in number and have myosin heads along their entire length. In fact, the ratio of thick to thin filaments in smooth muscle is much lower than skeletal muscle. In smooth muscle, it's about 1 to 13, and skeletal muscle, it's about 1 and 2. So thick filaments have heads along their entire length of the smooth muscle, which means that they're still strong, they're just not as numerous. Now, the troponin complex is actually missing in smooth muscle. And so it's kind of weird here where there is no troponin to bind with calcium. Rather, calcium interacts with another protein called calmodulin, which has a very different role than troponin within the sarcomeres of skeletal and cardiac muscle. Now, thick and thin filaments are arranged diagonally within smooth muscle, which allows for these cells to kind of contract in a corkscrew manner. And because they're diagonally, there's not really a sarcomere, which means that smooth muscle cells don't have striations. Instead, what we have is this intermediate filament dense body network, which is a lattice-like arrangement of intermediate filaments that resist tension. And dense bodies are kind of like the anchoring sites, like the Z-discs, for these intermediate filaments. And so that during contraction, the sarcolemma actually starts to bulge outward between these dense bodies because the cell kind of bunches up and it makes the cell look kind of poofy. So here's what a contracted smooth muscle cell would look like, where basically the myosin filaments would pull on these intermediate filaments linked up with the dense bodies, which allows for the whole cell to kind of bunch up and squeeze in a corkscrew manner. Now, in terms of the three major types of skeletal, cardiac, and smooth muscle, we're just going to compare and contrast these muscle types here. We know that skeletal muscle is attached to bones. It's under voluntary control, and the cells are these long, straight, slender fibers that are striated and contain many nuclei. That differs from cardiac muscle, which is highly branched. The cells are short, and it's under involuntary control, but also has striations like skeletal. Smooth muscle cells are short like cardiac, but smooth muscle cells don't contain sarcomeres, so there are no striations here. And so we also find that in terms of the packaging of skeletal, cardiac, and smooth muscle, skeletal muscle is more highly packaged than anything similar to like cardiac or smooth. In fact, cardiac and smooth really just have a lot of endomysium that packages these cell types. Now, we do find myofibrils that are arranged in sarcomeres in both skeletal and cardiac muscle, but smooth muscle doesn't have sarcomeres. Rather, its actin and myosin filaments are arranged in those crisscrossing patterns that we saw earlier. You do find sarcoplasmic reticulum in all three muscle types, both skeletal, cardiac, and smooth, but the sarcoplasmic reticulum is very scant within smooth muscle. Rather, in cardiac and smooth, it's more abundant. In fact, the sarcoplasmic reticulum in skeletal and cardiac muscle basically surround the myofibrils, but there are no myofibrils in smooth muscle. In terms of the sarcoplasmic reticulum network, you find it all over, in skeletal, cardiac, and smooth, except for in smooth muscle, it's a little bit less of the cell volume here. You don't find gap junctions in skeletal muscle. So skeletal muscle can't communicate between cell to cell via gap junctions. But that does differ from cardiac and smooth, where there are gap junctions that connect these cell types together. And that actually allows for these cells to transmit their electrical impulses from cell to cell. So cardiac and smooth muscle can actually spread their electrical currents all throughout the muscle. Now, in terms of how these cells are excited, skeletal muscle is excited by the neuromuscular junction. Cardiac muscle cells can be stimulated, but they're more rarely stimulated by your nervous system. Smooth muscle is stimulated by neuromuscular junctions, but it's different because we actually have calveolae, which are those expanded blebs of a nerve fiber. In terms of where these nerve fibers come from, your skeletal muscle cells are actually activated by the somatic motor neurons of the voluntary nervous system, but both cardiac and smooth muscle actually are stimulated or inhibited by the autonomic neurons, which are under involuntary control. In terms of the source of calcium for skeletal and cardiac muscle, you find that it comes from the sarcoplasmic reticulum, except cardiac muscle can also get its calcium from the extracellular fluid because it's part of the cardiac action potential. And smooth muscle also gets its calcium from both SR and the extracellular fluid. Because skeletal and cardiac muscle both have myofibrils, they have the whole actin, troponin, tropomyosin complex. But because smooth muscle doesn't have myofibrils, it doesn't have troponin either, it's got its own separate mechanism. What's kind of cool about cardiac and smooth muscle is they actually contain their own pacemaker cells, which means they can generate their own action potential spontaneously, but skeletal muscle can't do this. And so skeletal muscle can only be excited. Like, you can't prevent skeletal muscle from contracting, you can only excite it. You can't inhibit it from contracting. In fact, the only way to prevent a skeletal muscle from contracting is just to not stimulate it. But that differs from cardiac and smooth muscle because cardiac and smooth muscle can be both either excited or inhibited by hormones and neurotransmitters. In terms of the twitch speed, we see that in skeletal muscle we have fast twitch and slow twitch fibers, whereas cardiac and smooth are slow to very slow, and this relates to the speed of the myosin ATPase. Skeletal muscle cells don't have rhythmic contraction. They're just going to contract once in one quick twitch, whereas cardiac and smooth muscle can actually generate their own rhythms because of their pacemaker potentials or pacemaker electrical activities. In terms of metabolism, we see that skeletal muscle cells can be anaerobic, whereas all three muscle types mainly rely on aerobic respiration. So what we're going to do now is really get into the mechanisms of smooth muscle contraction. And we find that smooth muscle contraction is more slow and synchronized. And this is actually because of these electrically coupled gap junctions where action potentials can spread from one smooth muscle cell directly into the next. And what's cool too is that some of our smooth muscle cells are self-excitatory and therefore can depolarize without any external stimuli, like without neurotransmitters or hormones. These cells can actually generate their own action potentials at their own pace. And so that's what we call these pacemaker cells. And the rate of intensity of contractions can be modified by, neural or chemical stimuli. So in terms of contraction of smooth muscle, it's similar to skeletal muscle in a variety of ways. We have actin and myosin. The trigger is calcium. We need ATP. And the contraction stops when calcium is no longer available. But that's the end of where they're similar. What we find is that smooth muscle actually differs quite a bit because most of the calcium in smooth muscle contraction actually comes from the extracellular fluid. Very few of that calcium actually comes from the sarcoplasmic reticulum. Also, that calcium binds to a protein called calmodulin. There is no troponin in smooth muscle. And so that what happens is calcium activates calmodulin, which activates this myosin kinase. Myosin kinase phosphorylates the myosin head, which then activates the myosin head and allows it to form a cross bridge. So stopping smooth muscle also requires calcium removal. In smooth muscle, calcium is pumped back into the sarcoplasmic reticulum, but it's also pumped out of the cell. So we need active transport of this calcium into SR and out of the cell. And dephosphorylation of the myosin head needs to occur as well in order to relax the smooth muscle. So to see all these steps of smooth muscle contraction laid out, we have about a 5-step process here, where voltage-gated calcium channels open when the action potential rushes by. Calcium flows into the cell. That triggers what we call calcium-dependent calcium release, which we get more calcium from the sarcoplasmic reticulum. That calcium now floods the inside of the smooth muscle cell and interacts with a protein called calmodulin. Calmodulin activates another protein called myosin light chain kinase. And what the myosin light chain kinase does is puts a phosphate group onto the myosin head. And that phosphate group actually comes from ATP. And what this does is actually energizes the myosin head to be able to form a cross bridge with actin. That way it can participate in the power stroke. However, this process is much more slow, which explains why the smooth muscle contractions are slower than, let's say, skeletal or cardiac muscle. Now, smooth muscles are actually very energy efficient. Not only because they are slower to contract, but we actually can use less ATP for these muscles to contract. And myofilaments can actually latch together to save energy. And what's pretty interesting too is that smooth muscle maintains a constant steady state of contraction, which we call smooth muscle tone. And most of this ATP from smooth muscle contraction comes from aerobic respiration because it's slow enough that you won't really run out of ATP. Like you can rely on aerobic pathways to maintain smooth muscle contractions. Now, in terms of regulation of contraction, it's controlled by nerves, hormones, and chemical changes. Neuronally, we have neurotransmitters that bind to receptors on the smooth muscle cells that can cause graded potentials, which may or may not cause an action potential. These action potentials cause the release of calcium from the sarcoplasmic reticulum, which can lead to a muscle contraction. But what's cool about smooth muscle is that its response depends on the type of neurotransmitter that's released, so that you don't always get the same response. Because we can get different types of neurotransmitters and different chemicals to bind to receptors, we get varying responses by the smooth muscle because one neurotransmitter actually might have a stimulatory effect on that smooth muscle, but it might also inhibit that smooth muscle if it's a different neurotransmitter. So we also have hormones and other local chemicals that can interact with smooth muscle cells, and these can cause smooth muscle to depolarize in a way that allows for muscles to contract. But smooth muscle cells can also respond to factors like hormones, carbon dioxide content, pH, as well as low oxygen levels. And this makes smooth muscle more dynamic than, let's say, cardiac and and skeletal because it can respond to a variety of different stimuli. Now, smooth muscles can respond to both neural and chemical stimuli, which kind of aids in the dynamic capability. Smooth muscle can also respond to stretch, which is interesting. So one of the responses here we can get is a stretch relaxation response, where smooth muscle responds to stretch only briefly, but then adapts to a new length. So this allows for smooth muscle to contract on demand and enables organs like your stomach and bladder to basically temporarily store contents and then contract only when necessary. We also see that there's length tension changes in smooth muscle, where smooth muscles contract between half and twice of its resting length, and this allows for organs to have huge volume changes without becoming flabby when relaxed. So for instance, your bladder can stretch to quite a bit and hold quite a bit of urine, and when the bladder is empty, it's not going to be flabby when it's empty. Rather, because of the muscle tone and smooth muscle, it maintains this contracted state that allows the organ to, maintain some structure and shape and not be flabby when it's relaxed. Now, in terms of the types of smooth muscle, we find that there's unitary and multi-unit smooth muscle. And this is going to vary depending on, different organs. So it can vary based on the fiber arrangement, innervation, and responsiveness. So unitary smooth muscle is commonly referred to as visceral muscle. You only find this in hollow organs, except for the heart, and it possesses all common characteristics of smooth muscle, like opposing circular and longitudinal sheets. It's innervated by varicosities and exhibits spontaneous action potentials, which are the pacemaker potentials. And these cells are electrically coupled by gap junctions so that currents can spread from one cell to the next. It can also respond to various chemical stimuli. Now, multi-unit smooth muscle is located in large airways of your lungs, larger arteries, as well as the erector pili muscle and the iris of your eye. The iris is the pigmented part of your eye, and it controls the diameter of your pupil. Now, you find that there's very few gap junctions, and spontaneous depolarizations are rare. And for this reason, multi-unit smooth muscle then depends on chemical stimuli from basically, the varicosities we saw earlier, and these actually form motor units. And because of the fact that these cells are more independently controlled by motor units, it allows for a more directed and concerted, more precise form of muscle contraction than you'd find in the visceral or the unitary smooth muscle. And so what we find here then is that it's more precise, which makes sense why you'd find it in places like the iris of your eye. Now, it's different from skeletal muscle because, like unitary smooth muscle, it's controlled by the autonomic nervous system and hormones, which means that although multi-unit smooth muscle is regulated very similarly to skeletal muscle because there's motor units, it's still under involuntary control.